My name is Caroline Obert. Today we're going to discuss how Loopseq technology can be applied to targeted long-read transcriptome sequencing. This 2021 Communications Biology paper is an in-depth analysis of Loopseq chemistry as applied to transcriptome sequencing. We'll discuss over the next few slides how the University of Pittsburgh researchers determined that the methodology not only yielded highly accurate full-length transcripts with a 0.01% per base error rate, but that it is also able to be used to evaluate known and novel isoforms to track disease progression in metastatic cancer samples. These latter experiments could not have been conducted using short-read or other less accurate long-read technologies. As a part of Loopseq chemistry, unique molecular identifiers are first attached to long first-strand cDNA molecules using reverse transcription. While whole transcriptome sequencing can be performed, the investigators in this study incorporated an optional step by choosing to focus on specific genes and employing probe capture targeted enrichment. Then, using proprietary chemistry, clonal copies of a given molecule then have its UMI randomly distributed along the full-length sequence. These UMI-distributed molecules are then subjected to NGS library preparation and short-read sequencing on either an Aviti or another third-party sequencer. During sequencing, the UMIs are read first, followed by the long-read sequence that is immediately adjacent. Finally, the reads are binned by their respective UMI sequences, and the long molecules are reconstructed using a de novo assembler in a manner similar to genome assembly. A key advantage to the Loopseq chemistry is the introduction of known sequences on the 5' and 3' adapters. Incorporation of predefined sequences enables researchers to determine whether a synthetic long-read molecule is partially reconstructed or truly a molecule that has a previously unknown transcription start site or transcription termination site. By evaluating the ERCC RNA control, researchers found that approximately 83% of the transcription start sites and 69% of the transcription termination sites on the synthetic long-read molecules were at expected positions. While they did note that roughly 13% of the molecules had a transcription start site downstream of annotated sequence, this phenotype can occur if the cDNA molecule was prematurely terminated during reverse transcription as a result of degraded RNA or nonspecific terminal transferase activity. Similarly, while 16% of molecules had a transcription termination site within 5 nucleotides of the annotated sequence, a mispriming phenotype can be seen if the 3' end of the poly-T oligo overlaps past the correct poly-A start site, which causes an offset in the frame alignment. In a separate experiment, the accuracy of transcript quantification was specifically examined by comparing observed synthetic long-read ERCC molecule expression abundance to expected abundances. The image on the left shows the correlation between expected and observed expression abundance for all synthetic long-read molecules. The depicted correlation is comparable to previously published synthetic and native long-read technologies. The image on the right shows that when we focus on molecules at least 700 base pairs in length, we achieve tighter correlation between observed and expected transcript abundances. It is important to understand the amount and types of errors that can be attributed to the Loopseq assay versus that which is inherent to sequencing. The figures above show that substitutions are generally random regardless of assay or platform. Both the Loopseq synthetic long-read data and previously published third-party platform short-read data indicated that insertions and deletions occur near very specific homopolymer stretches. As similar data was evident irrespective of assay or platform, we can infer that the errors are neither a result of the Loopseq assay nor the platform, but rather from introduction during reverse transcription. Short-read sequencers innately have lower error rates than that found with native long-read sequencers. Another advantage of the Loopseq technology is that it utilizes the systemic advantage and augments it by incorporating the randomly dispersed UMIs to create uniform coverage throughout the long-read molecule. This uniformity further reduces error rates by circumventing low coverage regions, which inherently are more error-prone. In the figure above, RNA-seq library preparations were evaluated using Illumina short-read sequencing, PacBio and Oxford Nanopore native long-read sequencing, and Loopseq synthetic long-read sequencing. Loopseq had error rates that were at least 2 orders of magnitude lower than PacBio or Oxford Nanopore, and an order of magnitude lower rate than Illumina short-read sequencing. As suggested in the previous slide, the slightly elevated error rates across the board are attributable to the conversion of RNA to cDNA. When DNA molecules, shown in light blue, are sequenced, the indel errors found in Loopseq are reduced to 1 in 220,000 bases. The authors of the paper further assessed the robustness of Loopseq technology by preparing and sequencing 3 pairs of primary colon cancers and their corresponding metastatic tumors, and focusing on 2,193 specific genes of interest. Because of the resolution that can be obtained using the Loopseq chemistry, both gene and isoform level information could be obtained and is presented above. In the figure on the left, we see that using just differential or gene expression, researchers could not distinguish between primary cancer and metastatic samples. However, differential isoform expression clearly formed well-defined clusters between normal tissue and primary and metastatic tumors. The image above indicates that capitalizing on Loopseq's ability to detect isoforms will further enable researchers' insights into the transcriptome, especially in the area of oncology. The top image shows that in the absence of data pertaining to isoform alterations, a focused study on differentially expressed genes cannot resolve normal tissue from cancer tissue. The inclusion of the isoform information in the middle images, or even just the focus on genes with isoform alteration in the bottom images, however, shows a far more distinct segregation resolution. Additional investigation into the Loopseq reconstructed isoforms revealed several interesting findings. First is that certain single nucleotide variants, SNVs, were primarily found in specific isoforms rather than being evenly distributed. These specific isoforms had significantly different expression patterns than equivalent wild-type sequences. Secondly, when isoforms with uniformly high or low SNV rates were compared to their metastatic counterparts, the hierarchical clustering, displayed in the figure on the left, showed distinct separation. This finding could be utilized to allow investigators to track disease progression. Finally, utilizing Loopseq chemistry allows for the discovery of fusion isoforms which would otherwise have been impossible to detect. In the image on the right, we see that 4 of the 6 colon cancer samples evaluated displayed previously undetected fusion isoforms. We hope that this discussion of long-read transcriptome sequencing has provided you with new information about how this revolutionary technology can be used to address questions that were previously thought to be unanswerable. If you're interested in learning how Loopseq technology can advance your studies through long-read sequencing, please visit us on the Element Biosciences or the Loop Genomics websites.